Hi everyone, I'm Amelia from the University of Granada. The title of my poster is Unsupervised Protein Embeddings Outperform Handcrafted Sequence and Structure Features at Predicting Molecular Function. The automatic function prediction task using machine learning algorithms has shifted from using handcrafted sequence features to automatic learning these features. However, deep models require a large amount of labeled training samples, which are not available for the task of function prediction. On the other hand, millions of unlabeled protein sequences are available in databases as UniProt. These can be fed into a supervised model that learns a general protein representation to be used in other protein-related tasks. Here, we structure protein embeddings using a recent unsupervised model based on the language model ELMO, which were trained on millions of protein sequences. For the protein function prediction task, we combined protein sequence and structure information. We first compared the ELMO embeddings, both amino acid level and protein level, to two handcrafted sequence representations, the one-hot encodings of the amino acids and the K-mer count. We also structured several features from the 3D structure, including the secondary structure and backbone angles, which we compared to the pre-trained deep fold and contact map representations. The contact map can be viewed either as an image or as the adjacency matrix of a graph. In the experimental setup, we compared these protein representations using several prediction methods. The KNN, logistic regression, and the multi-layer perceptron classifiers work on the protein level features. The 1D-CNN and GCN models work on the amino acid level features. And the GCN and 2D-CNN models can process the contact map alone by viewing it as a graph or an image, respectively. We first showed the molecular function performance using a dataset with protein structure from the PDB. The ROC-AUC results on a 30% identities test subset show that the ELMO embeddings, which are in orange, outperform the rest of protein representations, and they are closely followed by the pre-trained deep fold features, which are in green. The contact map information provides a performance boost in the GCN classifier when using a simple representation as the one-hot encodings in blue. Also, the contact map alone in gray can predict molecular function better than the BLAST classifier, but worse than the pre-trained ELMO and deep fold representations. The superiority of the ELMO embeddings compared to other representations is also seen when evaluating the biological process in the left and the cellular component in the right ontologies. To analyze the contribution of the contact map in the GCM model when the ELMO embeddings are present, we change the adjacency matrix from the contact map to a random matrix and identity matrix. As you can see, the performance when using the ELMO embeddings is the same for the three adjacency matrices, but it gets worse for the one-hot encodings when changing the matrix. This suggests that the main performance of this GCM model is given by the use of a powerful node representation and not from the contact map. We then analyzed the performance of the models at different levels of term specificity. We used a larger dataset with sequences from SwissProt, evaluating 441 molecular function terms. The figure in the left shows the differences in ROC-AUC between the 1D-CNN using one-hot encodings and a simple KNN using ELMO embeddings versus the path length to the root in the goal graph. We found that the 1D-CNN model performs better for more specific terms. Other models that use ELMO embeddings, as the MLP shown in the right, had a better performance across all levels. This shows that these unsupervised ELMO embeddings contain a general function description, but supervised training is needed for the more specific terms. Finally, we perform experiments on the CAFA3 dataset, using several classifiers working on the ELM embeddings, and the CNN of the decode plus method, which uses one-hot encodings. If we compare with the top-ranked method in the CAFA3 challenge, our MLP method would have ranked 5th out of 146 methods. Also, a simple KNN using ELMO ranked 44 for biological process and 10 for cellular component ontologies. In summary, we saw the superiority of pre-trained features over handcrafted protein representation at the task of protein function prediction. The language model ELMO is useful to predict the protein function for the 3 ontologies. Also, there is not a performance boost when adding structure information to the ELMO embeddings, which is helpful when combined to a simple one-hot encoding representation. And the ELMO embeddings contain information about general functions, but further supervised training is helpful for the more specific terms. This concludes my presentation. I would be happy to answer any questions in the chat or by email. You can also check our preprint in the QR address. The links for the preprint and project GitHub are included in the posted PDF. Finally, I would like to invite you to attend my presentation on the function track on Tuesday, July 14th at 3:20 p.m. to hear more about this project. Thank you so much.